Netcasts you love.
From people you trust.
This is Twit.
Bandwidth for Futures in Biotech is provided by Cachefly at cachefly.com.
This is Futures in Biotech, episode 64. The future is viral.
I believe that biotech is the next frontier.
Probably the greatest intellectual revolution that's ever taken place in man's history. DNA is the code for life. We're actually beginning to understand how life works, which I think is something that's mind-blowing in and of itself.
There was going to be a genetic component to aging.
How long was there the extension?
About 30-40% for humans. That would equate to something like 20 to 30 years.
How close are we to actually having a therapeutic?
Ballpark, 10 years.
It's potentially one of the things that will end up rocking the world the same way that people said, oh, the sun's the center of the universe, oh, this and that and everything. And now here's somebody who can come out and say, hey, look, here's how we compare it to our closest evolutionary relative.
Welcome to Futures in Biotech. I'm Marc Pelletier. Today, I thought we'd bring it down a notch. It's summer, it's hot out, it's really hard to think. So what I thought I'd do is bring five of my closest friend scientists into the room, and we'd talk about the hopes and aspirations of biotech. Where is biotech going? Over the weekend, I watched Blade Runner, and I realized that the movie was set in 2019 in Los Angeles, and that is approaching. That's only 9 years away, and we don't have our androids, or well, we have Android, but not the Android. So I wanted to get their opinions, and scientists are constantly thinking about these issues, yet the way the media portrays it is that, well, it's up to the politicians to really make decisions, and let's get a first-hand account, again, with what the mission of Futures in Biotech is, to get that first-hand account. But we're stepping out of the lab. So we're going to talk about issues like global warming, biofuels, genomics, proteomics, personalized medicine, gene therapy, molecular pharmacology, vaccines, imaging, prosthetics, bionics, brain-machine interfaces, tissue engineering, artificial organs, synthetic organs, farming and biotechnology, GM plants and animals, stem cell research and adult stem cells, human embryonic stem cells, and the biology of aging. Is that a good list? Let's see how much we get through. So let me go around the table here and introduce. I'll start off with the folks that are in the Aromics studio. We have on our left here is Dr. George Farr. He's Vice President of Biochemistry and Biophysics at Aromics LLC, and he's an adjunct professor in the Department of Physiology and Biophysics at Case Western Reserve University. Welcome to the show.
Hello, everybody.
To his right, we have Marc Griswold, who is an associate professor in the Department of Radiology at Case Western Reserve as well. And well, he's an associate professor at a number of departments and one hell of a physicist. So I've invited him on the show to talk about some of the great imaging stories. Welcome to the show, Marc.
Thanks for having me.
Next, let's switch over to our remote lines. Do we have Dave Brodbeck?
Right here.
He's associate professor and chair, Department of Psychology at Algoma University in Sault Ste. Marie. Yep. And I think he's the head of science there.
That was, yeah, I was, but that was an interim position, and the person came back from sabbatical, and I'm so happy.
Cool. Now you can get back to research and all kinds of great stuff, and the science of mind.
Yeah, exactly.
Next, we have Vincent Racaniello, who's a professor of microbiology, host of This Week in Virology, and he's a professor at Columbia University. Welcome to the show, Vincent.
Hey, Marc. Good to be back.
Yeah, Vincent's the guy who created the first mammalian virus from scratch, right? He did what Craig Venner did recently. He did it 30 years ago, which is brilliant.
With a lot less fanfare, Marc.
Yeah. Did you even leave your bench, or did you just continue working? I'm sure it hit it was a great project.
Actually, it was in the New York Times. Yeah.
Oh, cool. So no, it was a breakthrough. It was the first synthetic non-living organism. Living? I don't know. We'll
some
talk
people
about
think
that.
viruses are living, but let's say it was the first synthetic organism. Sure.
Cool. It's an honor to have you on the show.
Thank you. Thanks.
And lastly, we have my PhD mentor, Dave Thomas, who's professor and chair of the Department of Biochemistry at McGill University in Montreal, Canada. He's also the Canada Research Chair in Molecular Genetics. Welcome, Dave.
Hi, Mark. Nice to see you.
I've wanted to have you on the show for a long time, and it's a great opportunity. I think this is a really great way to get people to know the Dave Thomas scientist. This is I'll warn people. He's got some great ideas, and they're pretty quirky. We're gonna get into them. Hey, so let's start off with global warming, a topic we know nothing about because we're biologists and physicists. And but we do have a Marc Griswold here in the office has a business with his family, and they're working on biofuels. So he might have an insight. First of all, let's go around the table and quick vote. Do we believe in global warming?
I believe in global climate change.
All right.
How about that?
George?
I believe in peanut butter. I believe in Peter Pan.
Dave? Dave?
Yeah, I'm always best at talking about things I know nothing about. So I'll say, yeah, I'm hoping for global warming. I live in Canada, and we could do with some.
Spoken like a true Canadian. And Vincent, what are your thoughts? Are we to trust every weather service, National Weather Service across the globe, that their science is correct? Would you want them to trust us?
Absolutely.
On matters
they just have to read the peer-reviewed literature, right, and get the truth. So, I mean, their data looks pretty good. I think I'm all for it. I believe it. I think it's getting hotter, and we need to do something about it.
Cool. So will biotech be able to address that? And so tell us a little bit about what you guys are doing, if you're allowed, without losing any intellectual property here. Marc?
I mean, I think that we can talk in general. I think that there's a lot of motivation to start growing our energy, that there's clearly limited amounts of oil. I don't know that we want to be drilling in the Gulf anymore. I don't think that's the solution. So it's got to come from somewhere. We're either going to all sit down and become cave people again, or we've got to come up with some other way to do it. And growing it is something that seems pretty reasonable. The problem comes, how do you not compete with food? How do you not make the greenhouse or climate change problems even worse? And those are not trivial problems. And I think that, it's going to come down to how can you grow the best or the most stuff per acre. And clearly, just like biotech has had huge impact in our food crops, it's going to happen in energy crops as well. I mean, there's just the fact that there's a term called energy crops. If you Google energy crops, you'll find something. Just the fact that something like that exists should be an indication of how this is going to go forward in the future.
Do you need biotech for this? Is it going to be the engineered crop that's going to be the most productive?
It has to be, right? I mean, we know this from food already. We know that, none of us eat, at least we're all in America, North America at least, none of us eat non-genetically modified food. I mean, maybe it's not, that they've inserted a gene to prevent the stuff from getting killed by Roundup, but every bit of corn and soybeans and rice and everything that we eat has had some sort of genetic modification to it through breeding or whatever. But sure, that's all biotech in my mind. And then when you add in, Roundup resistance is just one thing that you could add. You could add, the ability to break down quickly so that the post-processing would be good.
Is the equation really flat, though? I mean, if you're going to grow plants, you're going to fix carbon, which is fantastic. Then you're going to use it and release it back into the atmosphere. How about the energy used to grow those crops? Is it carbon neutral, or is it a little bit on the? Does it need supplement?
So this is a philosophical question, I think, and the way I think about it is this is long-term storage. This is a long-term energy storage of solar energy, right? If you could put a panel out there, a solar panel, and have it collect energy and store it somehow, that would be great. We don't have that right now. So I think that these plants that are going to get planted are storage devices. They're going to suck in the energy from the sun. They're going to store that in the leaves and in the fruit or whatever is going to be made, and then we're going to reprocess that into some other chemical form. I don't think that you need energy in the system to get energy back out, and in my mind, that's where it's going to come from. And I think that you can do the processing that way too. If you use your renewable energy sources like wind and solar just to run your processing, I think that it's potentially a way that we can do this and still maintain a true carbon neutrality and still live our lives.
Should we be looking towards, in terms of chemical energy versus nuclear energy? Okay, so you're a physicist as well, and you've invested a lot of time and effort into biofuels. What are your thoughts versus the nuclear option for unlimited source of power? The power of these
the nuclear option. Let me put that, Mark.
It's like, wait a second, dude.
Is it the nuclear option? Is he going to press the
the
button?
rest of the world, and we all have energy, right, Mark?
But hang on, let's think about this for a second. I mean, biofuels are sort of a stopgap, right?
Are they?
It kind of carries us over into a future where our primary energy needs are taken care of by some other means.
What is that?
Like nuclear energy, a combination of solar energy, things other than biofuels.
So here's the thing that you got to work out. So you got to figure out some way to get a car to drive and not have it be a hazard, right? So I'll confess, a hybrid car, and my fear of having a hybrid car is the battery. If you get in an accident and that battery gets damaged, you are at real health risk, which is not a problem that you have with a normal car. A normal car, you get in an accident and you can build a cage, But
if the gas tank gets damaged, I mean, things going to explode.
It's a small fraction of real accidents. I mean, most.
But can't you protect a battery in such a way that?
Yes. Now, this is the question. So now you've got to build an armament around this battery, right? Which is going to add weight and add everything else. And you get to a certain point where you have to carry the battery becomes the dominant weight that you're carrying. So if you look at most of the modern electrical cars, it is the dominant weight that's already there. And so when you start trying to increase that the energy storage thing that you've got there, you run into a situation where you can't build a big enough thing, really.
Well, Mark,
right? You're going to have to refuel.
Let me
somewhere
interject here for a sec.
It's not your show anymore.
Okay, but here's the deal. Will you be able to drive a warp core? We need a warp core. Warp engine. Will you be able to drive it with biofuels, right? Will chemical energy be the powerful? You have MRIs. You were talking about MRIs on what, episode 50 or 52?
51.
Yeah.
51.
Biological fuel.
Will you be able to use a biofuel to run or drive your MRI machines that you run daily? I mean, those are enormously.
So I guess my whole discussion here comes down to energy density, right? You've got to have some energy per volume, and you've got to be able to have all of the conversion equipment fit in that whole thing, right? So you can take the biofuel grass and everything else that we've got is horribly not dense enough, but we can, through pretty reasonable processing, turn that into something that's denser, right? So gasoline's pretty good. It's a liquid. You can burn it almost 100%. It has 85% efficiency in the engines. That's all not bad. Do you even need to, or can't you just convert your engine to burn little pellets of cellulose? Sure, you could, but
how efficient would that be, though?
Yeah, I don't think that you would beat the efficiency of today's gas.
Condensed, right?
Yeah,
sure.
Yeah, and so you brought up nuclear. So when you get into nuclear, it's really easy to convert that energy. That's true, but to protect it and to shield it and all of the required equipment around it, I just don't see that being feasible in a car.
In a ship, though, it is.
In a ship, absolutely, and that's why it's done today.
Yeah,
absolutely.
For sure, the military does it all the time.
Those aircraft carriers are fantastic. I mean, they go around the world, and an aircraft carrier, they talk about level of power, right? Okay, let's go to 80% power. Let's go to 50% power for the life of the ship. They never refuel. It's perpetual motion, basically.
Yeah, but that thing, it's a whatever, 1,000 square foot room where that core is, where you don't even go, right? And it's right. armored and everything else.
Well, some people go, right? Spock goes in.
No. I'm talking about the.
You shall remember, right?
Yeah.
Mark, that was a TV show. Just letting you know that there's reality as well. So Mr. Spock isn't real.
Okay. But we got it. Are we using? Okay, let's ask you. Are we using sci-fi to drive what we expect from biotech and from science? Not to this as a rat hole, but this show's about rat holes.
Well, I mean, I think that happens a lot. I mean, I think it's sometimes inadvertent, and sometimes it happens. I mean, like my phone here reminds everyone, of course, everybody said this, of a communicator and a tricorder. And was that driven by Star Trek? Who knows? On the other hand, I think a lot of people get into science based on watching science fiction programs, etc. I really do. I mean, I know that one of the reasons that I was interested in just doing science was because it just looked cool to find out how stuff worked, and it actually also makes it on TV look like it pays better than it actually does. So.
That's for sure.
Right. Well, so.
But no, I mean, you raise a really good point, David, because, if you think about, in the 19th century, we had Jules Verne writing all these great science fiction books. Most of the things that he talked about in those books are reality today. And, that kind of imagination and creativity that comes through literature is, I think, quite frequently, things that young people pick up on, that even older people pick up on, and begin to see what the possibilities are and see what do we have available today, and begin to make that technology reality.
So, hey, then just really back to the energy issue really quickly. It'd be really great to build a baseball field in the middle of a cornfield where that corn can be used for biofuels, right? Because how romantic is it to completely eliminate, the negative effect of energy from just pulling it out of the soil, right? We saw it in the Gulf, and we're seeing it. We're going to see it for the next 40 years. I mean, that's going to be disgusting. That one thing about the oil is it's just, even though it looks like the shores are clean, the ecosystem's been ravaged, and it's going to take 30 years, 40 years, maybe 100 years to rebuild an ecosystem back to where it was.
And to what extent are we going to pollute the planet in this blind lust to consume more and more energy, right?
Right. So if we can create farming or use farming that we currently have and focus it, or focus a small percentage of it or even half of it to energy, that's...
Well, I think one of the key things, just to put this out to this community, is that you can't just replace food with energy. There's just not enough food on the planet either. So we have to come up with, and this is where the bioengineering
Have
of
you
these
been
plants.
to Saskatchewan?
Yeah.
It's ginormous. It's huge.
Yeah, but it's not a great food production land, and that's exactly where I was going. That if you can engineer these crops in a way that they will grow in those marginal environments, marginal soil and marginal climate, that's going to be the solution. Not where you can taking corn out of our food system and putting it into energy. That's not a solution.
You're going to build an ecosystem.
Yeah, to
make it work.
Yeah.
Well, there's a lot of wheat in Saskatchewan, and it goes on for millions of square kilometers. So I think if you can use grass, cellulose, break it down, engineer an organism to really do it well, and then you might be able to make it economically viable.
But the bigger question is, will this solve the world's needs?
So can I just interject that at first it doesn't have to. But eventually, I think that you can imagine enough of this to work. Just take wind, for example. There's already studies from the USGS, I believe, that show that there's enough wind power in the U.S. to meet all the U.S.'s electrical needs. The problem is there's no grid and no storage to move it around.
That can be built.
Pickett's,
and he's right.
Scenario.
I mean, he's wacko. No offense to the man, but he's completely right.
You have to get the windmills out into
those infrastructures can be built,
and then find a way to get all the electricity into the grid,
and control it and move it around.
And that's probably somewhere where government can really step in and make that happen.
Absolutely.
Well, in Quebec, that's what happened in the 70s. Bourassa, to the chagrin of many, or to the
but that was
pleasure
hydroelectric, right?
Yeah, hydroelectric. They went in with major projects and built 5 or 6 major dams that have control the power or draw power from a drainage basin the size of Texas, right? So, and
if all that power got turned off right now, what would that do to the East Coast grid?
It'd be blackout.
It'd be done.
Yep. Yeah, this is Dave Thomas telling you, actually it did get turned off a few years ago, and if you remember, there were whole problems in New York, and the whole city was closed down for a couple of days.
I can remember
I haven't heard anybody say anything about actually the T. Boone Pickens plan to reduce dependence on foreign oil and just to conserve energy better. So what do you guys think? Are there biotech ways to actually conserving energy?
Conserve. Not do biotech, because I waste a lot of power in the lab.
No, we need to hear from David on this one, right? Because I think this is probably something where we need to kind of find a biotech way of convincing people that they need to behave differently.
Sounds like a grant.
It's a behavioral issue.
Yeah, I don't know. That sounds a little scary to me, actually. I mean, scary because if you want to actually sort of engineer people to, socially engineer people to be calmer. The thing is, the neat thing that you can do with changing the way people behave is you can make, if you take a look at something, it's very specific. You can't use great big broad strokes. Everybody, like for example, so sensitivity training never works. You don't tell people be nice to everybody because we're all humans. That doesn't work. What works is don't tell jokes about this racial group or you get fired. That works really well. That's how we don't smoke inside public buildings anymore. That's how we don't drink and drive anymore, because people said don't drink and drive. They didn't say alcohol, increases your reaction time, and also it's very specific. So if you went to very specific behavior, if you told people everybody put those CFL light bulbs in your compact fluorescent light bulbs in your house, everybody do that. And you're doing something good. And if you literally just said to them, do that, or you pick some other behavior, whatever the heck it may be, drive your car less. What they do in some big cities, even numbered license plates can go into the city today, odd numbers on another day. Those kind of things actually, that does work. Because if you tell people something specific, and that there's consequences one way or the other, then they'll do it. If you tell people everybody conserve energy, they're going to go, oh yeah, I agree. And they're just going to keep living the same way they are now. If you tell people put those light bulbs in because you're doing something good and it's simple, and there you go, that'll actually work. Like I said, that's how we got people to stop smoking cigarettes in buildings. I mean, it's amazing. I tell students it used to be that.
No, we taxed them. We taxed them out of the world. I can't see it on TV,
but people still smoke. But remember how we used to have. We're roughly the same age. Do you remember when you were an undergrad going to school and you went into the library and you couldn't bring food into the library, but there was a smoking area? I mean,
no, not that old. Sorry.
Well, only 45. There were study carrels at University of Western Ontario that had ashtrays in them.
Yes, you're right.
And that's gone. People don't drink and drive usually very much anymore because in the mid-'80s, someone said, stop it. And we all went, oh, yeah, you're right. They didn't try prohibition. We know that doesn't work.
Right.
All that leads to is Al Capone. But if you tell people, if you drink and drive, it stops. So
I still
this
think
is
an element of consequence. Like in the cigarette packages in Canada, if anybody ever saw them, they would show cancerous lungs on the front of the package. It was as big as the package in the picture.
Or the cancerous mouth, which is the one that everybody who smokes goes, oh, I don't want those. When the guy behind the person gets, no, I don't want that one.
The person with the jaw that's half ripped off because of mouth cancer. Those were, there was disincentives. I think taxing, if you drive up across the border into Canada, you see everybody drives small, little fuel-efficient cars up in Quebec, for the most part, because the gasoline's twice the price. Is that the way to go? I think if you get entrepreneurs like Marc Griswold here, who's growing an alternative fuel that becomes cheaper than the current fuel, for example, or you want to switch their behavior, there's economic incentives as well that can just, there's a better way to go. It's cheaper. How do you not do it? How about we switch this over?
Mark, I think if you told everyone that tobacco contains viruses, maybe they would stop smoking them.
They're
You know that.
scared of viruses, aren't they?
Listen, if you smoke a cigarette and then go plant your tomatoes, you're going to kill your tomatoes because it has a great pathogen of tomatoes in the tobacco.
You know, you could start that on a viral video even, and that would work. Is that biotech? Viral videos?
Viral videos. It's just accidental. It happens to be there. But missing from the discussion so far are the microbes, guys, because these have created much of what we have on Earth already, and we can manipulate them to do what we want. So, for example, we have an incredible hunger for protein. So we have cows. Did you know that in the multiple stomachs of cows there are bacteria that make methane? And all the cows in the world every day make 20,000 tons of methane, which is a worse greenhouse gas than carbon dioxide. So that we need to do something about that, because obviously the number of cows is only going to increase, and the problem, speaking of global warming, is going to get worse. So we have to learn how to manipulate our microbial communities in a way to get around this, and we can do it because they're incredibly powerful bacteria, yeasts, algae, sources of food, sources of fermentation products, even sources of fuel. So
maybe
I want to add
this
that.
is a stupid question, but aren't they eating the grass that's fixed the carbon? So the only way to grow a cow is to...
Yes,
but I guess
the cows, the bacteria in the cow gut digest the cellulose because the cows can't do it. They have actually bacteria, fungi, and archaea in their guts, and then the bacteria grow, and the cows actually eat and thrive on the bacteria, not on the grass itself.
But the cows themselves are carbon neutral because a cow will grow eating grass that's fixed carbon. So as our living organisms on life, I guess if we grow the population, it's equal, it's neutral. But using it to fuel a car is different. Or am I completely off base here?
Well, I'm just complaining about the methane being produced by the cows that we can't
use it though, right? Absolutely.
How can you capture it from a cow?
Capture it? Yeah, how do you capture it?
I've seen big domes over
pastures.
Not over pastures, over basically large. How to say it politely? Vats of cow excrement, right? Where they can use it to power 2 or 3 houses around the farm.
But most of the methane from cows isn't coming out of their part that produces the manure. It's coming out of their burps, actually.
Yeah,
really?
They belch it out. That's correct. Yeah,
almost all of it. Yep.
Don't smoke a cigarette.
In India, of course, they have these fermentation places where the cow manure is moved, and actually they power their cooking from methane produced in these concrete containers, so to speak, if you see it in some villages there. It's kind of a local sort of solution to a couple of problems. Of course, they don't keep cows the same reason that we do.
Let's switch over to genomics because we have Dave on the line here. So Dave Thomas, who's Canada Chair for Molecular Genetics. You've been doing genetics for a few years, and you've seen all the technologies evolve to what they are today. You probably have some great ideas as to where they should go. So we're switching it over here. Where do you see genomics? I mean, we're in a post-genomics era, right? We sequence the human genome. What's next?
Yeah. Well, it's strange you should say that. We were talking about it over coffee this morning, and so it's 10 years since we've had the sequence of the human genome. And the promise of that was always, we're going to identify a lot of diseases and come up with the solutions. So let's think. I can't recall that out of the knowledge of the sequence, we've got anything more than more problems, more genetic bases of disease, and we can recognize those. And if we think back, even pre-genomics, to things like sickle cell anemia, we've known the molecular basis of that for 50 years now, since Vernon Ingram, my guess, was looking at electrophoresis on starch gels of hemoglobin. So we've known about that. So the question is, where's the cure? And I think the answer is very complex. First, a lot of diseases that we identify in this way, there is no real market for them. There's no market push. I mean, that sounds bad, but I guess it's the reality of the society we live in. And other things like cystic fibrosis, we've known about that since before the genomic era. And again, no cure based upon the knowledge of the basic defect, to put it that way. So I think we're going to struggle with these things, at least the payoff for human health. What has worked really spectacularly and very exciting out of the genome is a kind of an archaeological genomics. So ironically, I think genomics is rewriting history and sociology. So I think, the sequencing of Neanderthal genomes, sequencing, I saw something the other day for Polynesian bones found in South America. I think these are going to be so exciting, and specific ethnic groups as well, showing their origins. I think this is going to be the sort of immediate payoff. Anyway.
Well, we've had Svante Pabo on the show, so I encourage people to Google Svante Pabo Futures in Biotech.
Yeah, well, that's a fabulous story. And I mean, these are really fascinating stories, and the impact, I think, is going to be in history and social sciences, and maybe understanding a little bit more about our origins. But, when you see something, as we saw recently, autism really has no no easily identifiable genetic basis, let's say that. I think it sort of tells us about what genomics isn't right now. I mean, we're all optimists. We all know that eventually there will be cures based upon our genomic knowledge, but it's a slow time in coming, and I'm not too sure why.
How about in diagnostics and personalized medicine? If you have markers, genetic markers, I mean, I can understand that if you're going to tackle human health, you've got accidents, you've got infectious disease, and you've got genetic disorders. Genetic disorders might happen 1 in every 3,000 people, whereas a heart attack will happen 1 in 5, right? Yeah. So our drive to cure 1 in 5, 1 in 7 for cancer, 1 in 20 for stroke is enormous. And those, do they have underlying genetic elements that we can now get to quicker, and then based on our genomic knowledge?
Yeah, so we've been doing some things on, for example, prognosis of cancer outcomes based upon RNA profiling, okay, on microarrays. And in fact, one of the latest students from my lab just defended a thesis this morning. And sure, you can do a prognosis, and sometimes these can be pretty accurate, maybe. They're just extraordinarily difficult to do, complicated, and to a certain extent costly. And even if you have a prognosis, what do? We know, without doing any genetics, and this goes back, I think, to Dave's point a few minutes ago about global warming. We know, without doing any genetics, that if you're overweight and you don't exercise, you're very susceptible to get diabetes and a few other diseases. So then that's good. Everybody knows that. It's been in the popular literature. There are lots of diets out there. So obesity is going down in North America, right? I don't think so. And what we need is Dave telling people lose weight.
Or else.
Well, hey, genomics is...
Or else is, I don't know, because we want them to buy more food, of course, to keep the economy going. Yeah, so...
Sure.
Well, Mario Capecchi, last episode of Futures in Biotech, he talked about one gene. He's studying HOXB8, right? Yeah. And they knocked the gene out of a mouse, and they could do that because of new genomic technologies that occurred in the past 15 years, for which he won the Nobel Prize. Yeah. And it found it modified behavior. Dave Brodbeck, do you think genomics has had an impact on your field of psychology? Is it merging right now?
Well, I mean, the biggest thing that's happening in psych right now, just generally, I mean, I do really sort of brainy biological stuff, but even in the more social science parts of psychology, people are really recognizing now the importance of sort of an evolutionary approach and evolutionary psychology in general. So people looking at us as animals, and really complicated, cool animals, but animals. So instead of thinking of us as some sort of something cool, I have a slide in one of my PowerPoint presentations that says, do you think there's any genetic effect on how you behave? And the next slide in capitals says, of course there is. What makes you think you're so damn special? And it's something that's really coincided really with I don't know if that's coincidental or not, but it really has had a big effect. It's happened at the same time, where once the genome was sequenced and that got out there, evolutionary psychology, which was this almost fringy kind of area of psychology, has become pretty much the mainstream approach to psych now, in everything but the sort of clinical areas, and that's coming now. So it has become a big thing. I'm all for it because I was trained that way anyway. So the world's catching up my PhD supervisor. But it makes me happy to think that, because I think that understanding that we're part of it, going back to the global warming thing, understanding that we're part of nature and that we're animals, I think really helps us get. I don't want to get too kumbaya-ish here, but it makes us realize that we're part of a system. The other neat thing is it shows us that you think about different ethnic, racial, linguistic, religious groups, the amount of genetic diversity in humans is so freaking small. You realize that there's not a whole lot of difference between all of us, which is also a cool thing. Again, as Dave said, just sort of almost sociologically, if you want.
How about proteomics? I mean, where are we at, Dave, with proteomics? I mean, you when I joined your lab, you were just finishing up a large proteomics project that identified key elements to which underlying mechanisms for all protein trafficking diseases, cystic fibrosis and Gaucher's and a whole slew of them, emphysema, and fundamental. Where's proteomics at? And first of all, what is proteomics? Before I use a medium or sized word, what is proteomics, and where is it at now, and how will that help us, if it will?
Right, good question. So proteomics is really defined these days by very sophisticated mass spectrometers that can measure very precisely fragments of proteins, the molecular weight, and establish them. There are a number of other technologies around that, but that's the main technology. And the hope there, clinically, is that we can, in some way, bring this into diagnosis, even into operating theaters, and establish the limits of tumors. We can identify mechanisms of disease, and that's probably been the best part of proteomics so far. And also that we can come up with diagnostic markers. For instance, like prostate-specific antigen, not so specific. Are there other markers that we can use for prostate cancer, breast cancer, and things like this? Preferably taking some bodily fluid and looking for disease markers in there. So
these instruments break it down, right? They break it down from here's a blood sample to the individual proteins, and then identifies the individual proteins based on what information? Genomics, right?
Yeah. It's like that. It's a little bit more complicated. You use a specific enzyme to cut up the proteins, and then you compare it. Actually, maybe this is one of the things that genomics has really done for us, because we know the sequence of many of the proteins in the human genome based upon just looking at their genetic code and knowing what the splicing variants, just to use a technical term, are. And so there you can just predict in silico what the different fragments of proteins are, and then know where the protein comes from. So yeah, so it's a little bit technical, but the sophistication of the instruments is just leaping ahead in terms of their sensitivity and their specificity. So sensitivity is very low amounts of proteins, and specificity is being able to identify very precisely in this case, the mass, the weight of those fragments of those proteins.
How long does it take to do a run?
To do a run? Seconds.
Yeah. Seconds. So we're talking tricorder
level. To analyze the run could be a long time, yeah. The software is not keeping up with the sophistication of the instrumentation.
Okay, Twit audience, let's get going on some proteomic software. We've got a very good sized audience of really great coders here, so let's
well, get to it, guys. We need things that work better, faster, cheaper. You know, all the usual things.
All of the above.
Let's turn the discussion over to viruses a little bit here.
Let's do that.
How is biotechnology, the current state of biotechnology and where it's going, affecting our ability to tackle viruses as a major human foe? HIV, where's it going?
There are a lot of ways that we use biotechnology. We use it to make new vaccines, for example. We can take individual proteins and engineer them, produce them in large quantities, and use them as vaccines. We can make antibodies, which can protect us. We can analyze the antibody response to understand how it can protect us. So, for example, we know that one of the biggest problems, of course, is AIDS. We know that some people make antibodies that can block infection, and now we can understand why. We can use biotechnology to identify the specific antibodies that do that. So there's a lot that we can do in terms of prevention, but what I think is maybe the most exciting is the ability to modify either humans or the vectors that spread viruses. So, malaria, to get a little away from viruses, but to stay on parasites, malaria is a huge global problem because there are mosquitoes everywhere. If we could modify the mosquito so that it would not support growth of the parasite, we could stop the spread. And you can do the same for viruses. Many virus infections are vectored by mosquitoes. And there's no question that we can modify these vectors so that they will not support virus replication. And that's a direct outcome of biotechnology. We know what genes to alter in the mosquito. The question is, one, would we be able to release a genetically modified mosquito? That's sort of a political question. Would we be permitted to do that? And secondly, can we design them so that they would replace existing mosquitoes? It's not clear to me that they would have any advantage, and so they might not spread in the global population. So I think that's a very exciting area to be able to modify vectors, because vector-borne diseases are huge problems.
How about rapid diagnosis of viral infections? I mean, now everybody's doing PCR in every medical office, right?
Yeah, PCR is wonderful, but it's not without its problems, and it's still a technology that is somewhat cumbersome. I mean, I'm looking for the tricorder, Mark, as we've said many times.
Sure.
Someday when we can scan a person and detect an infection immediately. There are now some very small handheld devices which can be used to look at, say, lymphocyte levels in people infected with HIV. These can be used in the field. They're sort of mini flow cytometers, if you will. And that depends on taking a little bit of blood, which isn't so bad. But I think what we really want is a bloodless solution. There are thermal scanners that can be used to see if you have a fever. So there should be ways to determine if you have some kind of a pathogen in you. So we're not there yet. The diagnosis right now isn't great. You can rapidly diagnose flu by a non-PCR method, by ELISA or antibody-based methods, but they're not reliable. In the end, PCR is still the best, and it's not a technology that anyone can do. I think we need to get to that point.
What about gene therapy? I mean, the promise of gene therapy has driven a lot of groups, including Oliver Smithies, who, created the genetically engineered mouse because he was trying to get a method of gene therapy, and it ended up being just a great way to genetically engineer a mouse. Is gene therapy going to be feasible? Are we going to get stable protein expression using specific viruses? Which viruses do we use?
Well, right now, it holds a lot of promise, but as I understand it, nothing's really materialized, right? Nothing's really been able to make it through the clinical trials.
No, it's not
entirely?
really true. There have been some successes. There have been some successes with gene therapy using. And right now, what we use, Marc, are mainly what we call lentiviral vectors. These are vectors based on viruses like HIV, which are not, of course, at all dangerous to be used in that situation, but they can carry a gene into you and stably or permanently put the gene into a cell. So a number of diseases have been addressed using this, not on a big scale. These are just experimental studies in a variety of different ways. And, for example, there are eye diseases that have been addressed successfully. There are some neurodegenerative diseases. So the lentivirus is the way to go. It works very well. I have no doubt that this will be feasible. So there are two things you can do. You can either put a gene in you using these viral vectors, or you can take cells out of you, put them in a culture dish, then put the gene in using the vector, and then put the cells back. So that's typically done with hematopoietic precursor cells. You can take them out of your blood. You can put a gene in them using a lentiviral vector and then put them back in. And most recently, that's been done to make people resistant to HIV infection. So I think there's a lot of promise there. We're going to see a lot of that in the next 10 years.
What happens if it becomes, such a routine methodology for gene transfer, right? And what, for example, Eric Kandel identifying CPEB5, is it? CPEB, which when overexpressed in neurons leads to an improved long-term memory potentiation. So we make a virus, everybody's twice as smart. Should this be done? Twice as smart. No, the ability to learn twice as
to my students, yes.
Do your students need it?
To be fair, Marc, I think he was doing that in a sea slug, right? In Pelletier.
No.
Pelletier, yeah.
So he started off understanding the molecular methodologies in Pelletier, which is giant slug for it's got giant neurons and easy to poke. Okay. But no, they did it in mice.
I think from Paul the octopus, we kind of know that cephalopods are pretty smart animals, right? He predicted every game in the World Cup recently. Yeah.
Did a good job.
Did he?
Yeah. It was every game that Germany played.
Absolutely.
Because he spoke German, right?
Yeah, I just wanted to interject about gene therapy. Yeah, it has some successes. There have been large areas where it has not worked. And maybe turning the model on its head is one of the things that will come out of genomics. And what I mean is, right now we're more or less focused on putting back a good copy of the gene that is defective. I think as we know more, as we get to know more about why defective genes make cells do what they do and then leads to illness in the organism, we may see other strategies of putting in small siRNAs to inhibit downstream effects. So you'll have a defective or a gene that isn't working properly or not in the right place, but we will be able to correct that and correct that level, maybe correct the degradation of that protein that's made by that gene by putting in small siRNAs when we know what the downstream effectors, the downstream effects of those genes are. So I think it would be, although there's been a lot of money spent on gene therapy, I don't think it's out the door yet. I think it's one day, and it'll come back in a different guise.
So siRNAs are basically anti-genes, right? It's a gene that binds to the message and then results in a degradation of the genetic information.
Precisely. Yeah.
Right. So anti-genes. So you've been working on a disease that people have looked towards gene therapy, cystic fibrosis, because it's clearly a gene defect. And
by some estimates,
what are your thoughts?
Which way? $800 million has been spent worldwide on gene therapy for cystic fibrosis. Yeah.
Is it just because it's not there yet, or is it really molecular pharmacology that's a drug that can?
Well, it's not there, unfortunately, because it doesn't work. I think the original ideas were just spectacular. People were putting a complete SCFTR gene, that's the gene where the mutation is, back into cells using an adenovirus, which infects the lung, and the major phenotype of cystic fibrosis is in the lung. And they were getting correction to a certain extent, or infection, and then correction of the problem by putting the wild-type gene back in. And it would work once, and then it wouldn't work again. They would not get therapeutically significant levels of the protein, for example. And there is a trial going on right now in the UK, which uses a synthetic gene, and we're going to talk about that later, with everything, all the bells and whistles that will make this gene be expressed very well, they hope. And also a novel method for not a virus this time, but a mixture of lipids that will mix with the cell membranes and get the DNA into the lung cells. Let's hope that this works. That's the last trial that I know of that's going on for gene therapy in cystic fibrosis. It's been very disappointing, I think, for the patients.
It's rough.
For the scientists.
Absolutely.
Yeah.
One of the difficulties for cystic fibrosis is getting across the mucus that's formed in the lungs of CF patients. So they're particularly hard to treat with a virus, right? Or could you do a systemic virus, bloodborne virus that would translate?
Well, CF patients, I mean, the lung is complex, and the problem of where the mucus is one of the problems. But CF patients are susceptible to viruses, to flu and whatever. In fact, maybe over susceptible. So the viruses can get in. I think the problem is really probably one of the more difficult ones, the ones that Vince just mentioned, you know, the retinotherapies and some of the blood disorders are going to be much easier than lung therapy in this way. And I think we'll learn a lot. Yeah.
I guess one of the problems is that the adenoviruses, of course, are what we call acute viruses. They go through a cycle of infection and then they're gone. And they're very different from the lentiviruses, which put their genome into that of the chromosome, into the chromosomal DNA. And so, and in addition, of course, the lung cells are turning over. So ideally, one would want to put a lentivirus vector in a cell, a precursor to a lung cell, so that you would always have lung cells produced with the gene in it. So there it gets very difficult, and that's the problem, and that's why the approaches, the lipid-based approaches are being tried. But I do think if we could figure out a way to permanently get the gene into the cell, it would be better than using these transient vectors like adenoviruses.
Yeah.
So maybe futures in biotech can actually, by combining Dave and who understands the molecular mechanism of the disease very well with a virologist who's incredible at making viruses, you guys got to work together, right? Co-write a grant, please.
Yeah.
There you go. So it sounds to me like the primary issue here is delivery, right? You really have to be able to get things there in a sort of a consistent fashion, and then you can possibly get the gene therapy to work properly. It seems like, you know, especially in the case that Dave was talking about, that you could be treated every so often. I mean, it could be not maybe looking at a single application to solve the person's problem, but it's something where you could have multiple treatments over the course of many months or years or whatever to give the gene to the cells as they come up in the lungs to correct the defect. I mean, there could be an ongoing treatment, and I'm sure the pharmaceutical companies would like that even better, because they would need you to come back for more drug on or more.
They would probably design it that way on purpose.
Yeah, for sure.
Well, wait a minute, guys. Do you think pharma doesn't like gene therapy because it's not a drug and you can't? I mean, lentivirus sounds like a great way to go, but sort of a dead end economic project, right?
Right. But if it's a situation where you can't get to the I guess in the case of the lung, is it epithelial tissue?
Yep.
That you can't penetrate down to the very bottom layer where the sort of progenitor cells are coming up from. If you can't penetrate all the way down with your gene therapy, then I think the pharmaceutical companies will say, great, no problem. We'll treat the ones up top that are causing the problems, and then as they slough off and the next round work their way in, we'll treat you again, and it'll be an ongoing therapy, for the rest of your life, and they can keep tapping you for more funds as you go.
Sounds like a business model to me.
Well, that's why he's VP of Aeromics.
No.
Okay, so
I'm just thinking that particular approach would be attractive to these guys. I mean, I think it would be very attractive rather than trying to fix it down at the bottom.
I'm wondering, should we talk? Well, Dave, you had a comment that Dave Thomas, you had a comment.
Yeah. Mm-hmm.
On the viral delivery system.
The latest trial, the one that's registered in the UK, doesn't use viruses. It uses a lipid formulation, I think, from a Japanese company.
Okay.
Yeah. So the virus that was being used was a defective adenovirus. So it would not replicate in the cells.
Okay.
There were a number of reasons to want to do it that way. I think the lentivirus way will be a much better way, and then certainly we use that for delivering shRNAs in the lab. But it's as you know, there are now a lot of people that are looking at other conventional chemical treatments, drug treatments, for CF patients and other protein trafficking diseases. So maybe these will work. But I think we all hope that a rational kind of gene therapy will be there for a lot of different diseases. I mean, it would be great. It's just we're not there yet. Yeah.
So, I mean, what are the real negative potential consequences to treating with some sort of a lentivirus type of approach, or with the liposome, or type of scenario that you're talking about, Dave? I mean, I thought that this was one of the biggest areas that the gene therapy has gotten into trouble with. It wasn't necessarily just that sometimes it worked and sometimes it didn't, but that there were real potential negative consequences to the delivery system.
Well, the lentiviral thing is, of course, as Vince can say, is if you're doing it in a human, there will be a lot, and that's what you're going to be doing it in. There'll be a lot of questions about safety. Where does the lentivirus insert and things like this. In terms of cells, the half-life of the cells that you're infecting and things like this. Those will probably have to be monitored quite carefully. I think companies would like to have just a formulation, something that's given, that's sprayed, let's say for a lung disease, that is sprayed into the lung, is nebulized and gets down to the right parts that are therapeutically relevant. There are probably going to be lots of different ways, as I said earlier, of tackling this problem. But the negative ways, the negative vibes that will come out will be probably less, I would guess, than we see for genetically modified foods. People don't mind having people walking around who are genetically modified. They have a suspicion of a genetically modified tomato or something. Okay.
Mm-hmm. One of the issues, as Dave said, is that the lentiviruses can insert into the wrong place in your genome, and they could conceivably cause cancer by doing that. And in fact, in a couple of cases in France, some recipients of lentiviral therapy developed cancers as a consequence of that. But that's not completely unpreventable. We can, if we can take cells out of people and infect them in culture and learn how to maintain them for a period of time, so we could study where the virus has gone into the chromosome, for example. We could say, okay, here it's all right, and we can put this back into the patient, or no, the virus has gone next to a tumor-inducing gene and we have to throw it out and start over again. So we can do that at some point. It requires a lot of understanding. Yeah, we have to understand what makes the virus go into certain places and how to make the cells live a long time so that we can do these studies. I think that's a reasonable approach to take, so that we can say, all right, for diseases where we can take cells out, put the virus in, then put them back in, we'll make sure that the virus doesn't go in a bad place.
So, assuming you could make a virus that you could completely control the level of expression of the genes that it was carrying. And I'll ask Dave Thomas to translate this one for the general audience. I'm preparing a virus. It's a lentivirus, global transfection, and I want to create an activated form of SIR2. And that's going to be, we're actually developing that in the lab to compete with Certus. Maybe you could translate to that audience, to the audience, and give them a perspective of what that would do.
So you want to put in sirtuin, you want to put sirtuin?
An activated sirtuin. Constitutively activated sirtuin. What would happen?
What would it do?
Yeah.
Presumably, Mark, I haven't seen your business plan yet, but presumably it will give you a lot longer life, and you won't have to drink red wine to get the compound. Yeah.
So this is coming, right? This is coming. Yeah, this is a virus that we could engineer.
Yeah. Yeah.
And make us all live to 150.
From what we deliver it to every cell?
That I don't know.
I think you'll have to push up the retirement age, so maybe
125.
So the sociological implications are maybe and are pretty big there, right? In theory, these things can be done. They'll be done in mice first, of course. They'll be done in cell culture.
By the way, it's done in mice. We already know that sirtuin activators extend the life of the mouse by 40%. Okay, done.
Yeah, but
not
so
genetically engineered, but a drug.
So does rapamycin, yeah. So, and so does calorie restriction, just to come back to something else.
I'm not doing that.
So, but in terms of gene therapy versus molecular pharmacology, right? I mean, you've got to deliver this treatment to all of the cells.
Sure.
So I think molecular pharmacology wins at the moment, because I can distribute a drug to virtually every cell in the body, if not every single cell in your body, but a gene therapy delivered to every cell in an adult organism. We're nowhere near that.
Yeah, good point.
Well, but the point being is, how does it change humanity if overnight the drug is FDA approved, and be it virus or drug, where is that going? What would happen if it happens overnight? Is it become a nutraceutical? Dave Brodbeck, maybe you have an idea on this. What would happen to everybody's mind? Would we go crazy? Was social chaos?
Well, you mean more than now? I don't know. I think that what ends up happening in these situations is the amazing, almost adaptability of people when things change like that. Now, we've never had a societal change that quick, but we do have things that are pretty fast. You think about historically things like the Industrial Revolution, right? People go from living, I was a cooper because my father was a cooper because his father was a cooper, back to when the Romans arrived. We go from, that was a pretty big change. Now, it would be nothing like the kind of change you're talking about. The thing that would concern me would be, who gets it and who doesn't, right? You want to think about ethics here. Who gets it and who doesn't? Is it
me and my kids? My kids, at least.
Exactly.
At least my kids.
Yeah, but that's the thing. Why do your kids get it and my kids don't get it? Right?
Well, we'll make sure your kids get it because you know me, right? Sorry. We just dropped the camera here, but...
Fine, we're fine then. But, I think that's I think, the biggest issue, and I think that you'd have I don't know that we've ever had that kind of change. I can think of, think of the Industrial Revolution is probably the biggest societal change we've had in a very long time, perhaps ever. And that still took a long time. That still took 50, 100 years. This is something that conceivably could happen exceedingly quickly.
So
right? Assuming
overnight, we could see the greatest societal change as a result of biotechnology. That is, that would be, and I'm not kidding. This Sertris was sold to GlaxoSmithKline for $700 million. If anybody wants to get some background on this, 4 years ago we did an episode with Leonard Garanti, who's the person who discovered these genes, and he describes the science, and he's the Novartis Professor of Biology at MIT. It's not flaky stuff. This isn't a cocktail that was derived from mixing some, worm guts and some green leaves. This is.
Hang on a second, Mark. I would just interject to you guys real quick that biotechnology, if we want to call it that, has already, in the history of mankind has already done things like this multiple times in the past. And the biggest example I'd like to point out would be the use of antibiotics. I mean, before World War II, the captain of death was pneumonia. And so many people, I mean, there were more people lost in World War I and all of the other previous wars to the effects of pneumonia, or in the case of World War I, the Great Influenza. But more lives were lost in those wars to various microorganisms that could be treated with penicillin or various antibiotics than were lost on the battlefield. And that's a huge change. Huge change to society.
Well, influenza killed 50 million people in 3 months, right? In the Northern Hemisphere.
Yeah. But just in terms of the toll of what antibiotics did. I mean,
but that's
enormous
50.
change.
Okay, that's 50 million people that could be saved with an antibiotic. That's an antiviral. But in the same situation where you have a bacteria that can infect and have a global pandemic, which could kill 100, 200 million people, 300 million people, we're talking about 6 billion people.
6.5 billion people. That's the difference. That's what I was saying.
Yeah.
I mean, there are, and you're totally right about antibiotics, but not everybody gets pneumonia or whatever. Not everybody gets dysentery. I don't know, whatever the heck you want to pick.
Wait, modern medicine changed though. It did change when antibiotics came in. Modern medicine changed. Now it's going to change again.
Yes.
I think we're on the cusp of that. That would be the biggest change ever. It seems to me.
The real key here, though, is that the antibiotics, the antivirals, our understanding of infectious diseases extended our life from the mid-30s to the 70s and beyond. And as a consequence, we have other diseases emerging. We have Alzheimer's diseases. We have late-onset cancers that we never saw before.
Yeah.
So any therapy that extends your life is going to have to take into account the fact that maybe other diseases are going to pop up. I think at the minimum, we should be able to deal with Alzheimer's before we make people live another 30 years or so.
Well, hey, there's the great...
It's a quality of life issue, right?
Guess what? That drug, the sirtuin activators made by Sertris, are actually being... FDA, or the clinical trials are on Alzheimer's and diabetes and cancer, because FDA does not approve, does not do clinical trials on death. Death is not a disease. But Alzheimer's, diabetes, and cancer are age-related diseases. So this drug, in fact, pushes those back. So hopefully, I mean, so far the clinical trials are showing this. So in fact, the drug actually handles that, or it handles that problem directly, right, by its nature.
Well, maybe it depends on how. I mean, I don't know if they're trialing it for Alzheimer's specifically or something else. It depends. They have to really do a very specifically designed clinical trial to answer that question. If that's the case, if you take this and it prevents onset of Alzheimer's, that would be terrific. But if it doesn't, then I think you have to back off. And, because you can imagine people will jump on this without understanding the consequences of living longer.
Yeah.
Right. It is a metabolic shift, right? It turns your metabolism into a hypermetabolism capable of how would I explain this, Dave? You're the biochemist, or actually you're the geneticist. So it basically switches your metabolism over to one that mimics caloric restriction.
Yeah.
Right. So
it's an HDAC inhibitor, histone deacetylase inhibitor. Yeah, of which there are many. Right.
Yep.
Yeah.
So I mean, this all gets back to what I like to call the first rule of pharmacology, which is that no drug has one effect. What are the other potential consequences to taking these types of inhibitors?
Well, one of the known side effects is your ability to without precondition being able to run a marathon, right? The mice are doing fantastic. You take a mouse, they can overeat, they're okay. They can get up and run 14 to 18 miles or so, they can
Marc,
do incredible.
The SIR2s, do they simply extend your life, or do they prevent the aging process? Do we know the answer to that?
They extend your life. What they do is they extend youthfulness, right? They give you a condition. If you're infertile, if you're a woman and you're taking the sirtuins based on the animals, you're expected to become infertile, but then be fertile at a much later age. When you get off the drugs, you now could potentially be fertile in your 70s.
What about issues such as decreased mental abilities, increased susceptibility to infectious diseases? Does the sirtuin have any effect on those processes?
I don't know. Those are what the clinical trials are all about. So we'll have to see. But is it fair to say that we're about to see a revolution? One at the scale of the last antibiotic, or the scale of how antibiotics change medicine?
I think it's fair to say that we're on the cusp of it.
I don't think so, because this is not going to be available to 9 billion people. It sounds to me that it's going to be extremely expensive.
I was saying too, yeah.
And it's not going to affect enough people. I mean, I think that these companies should make it a cure for malaria before they do that.
No kidding. Exactly. That would hundreds of millions of people would be saved. So
I think
so far it's working out, right? It all depends upon cost.
So go back to our first problem too. I mean, how in the world are we going to put 50% more people on the planet and survive?
Yeah,
I don't see that happening.
Well, no.
So in other words, it's not something as a society we should do.
No, I just think there's a lot of implications. Mm-hmm. There's a lot of implications.
Okay, you don't get it, Marc. Marc's volunteered not to take it.
It's going to be like Botox. It's going to be like Botox. Whoever can afford it will take it. The wealthy people will get it. They'll live longer. We'll have a dual society, or even greater reinforcement of a dual society, because it won't be available to everyone. It will be made because it will be FDA tested and approved. People will buy it, and a fraction of the population will use it, just like Botox. That's my prediction.
Just hope that they don't turn into zombies like in I Am Legend, right?
No, they'll be in glass buildings, and they're the ones that'll have the androids and see the sunrise and everything in 2019 when Blade Runner was supposed to happen.
God, that's a good film. That's why I got into genetics.
Yeah.
Did you try EMS mutagenesis? Right? They got it. They nailed it back in 1982. Right? We did EMS mutagenesis in Dave's lab, on yeast. So that was 10 years later.
Yeah.
Any last thoughts, Dave? Where do you see
Biotech taking us? Again, actually, I'm surprised I'm saying these sorts of things, is that the big effect of living longer is on society. And we're seeing it already in the age structure of societies. In Europe, there are a lot of baby boomers who are not being able to retire right now at the ripe old age of 55 or whatever, because the legislation is changing the retirement age because they cannot afford to let these people retire and hire somebody else in their place. So it's going to make in North America, we don't quite have the same age structure, but it could happen with things that are going to prolong life of people who are essentially, let's say, not as productive, putting more into society at that later stages of life as they were at earlier stages. So it would change sociologically. I think it would change a lot of things. And we'd need new systems and new structures to take care of these things, I think.
Ron Evans, who was on Futures in Biotech, was the individual who claims to have cloned the first gene. So Congress passed a law, and he was there in the lab at midnight waiting for them to pass the law to clone the first human gene. And by the morning, he has transformants in E. coli, and he had cloned a human growth factor. So sometimes laws are ready to be passed, and then we're ready to pounce, right, in the lab.
Yeah.
So it doesn't take very long. How about you, Dave Brodbeck? What are your thoughts? How is it going to affect your field?
Well, I mean, I think that this kind of stuff is going to affect everybody, no matter what. And I mean, the stuff we've just been talking about, I was just mentioning, and like Vincent said, the idea of this sort of increased stratification of society, if some of these things aren't available to most people. Now, some of them may be after a great period of time. I think that it affects a lot of these things, and we talk about this in my contemporary theory and psychology class. A lot of these things affect psychology because I think what's going to happen in psychology is that you're going to have things like, for example, for a lot of things that we think of today for using talking cures, I think we're going to end up using something we're probably not going to get to today, things like sort of personalized medicine and gene therapy and imaging to figure out what's wrong with them, and we'll just fix them. We'll give you the Marc Pelletier drug, and you'll be fixed. So I think that's going to happen. Right? I'm not saying there's anything wrong with you, but well, actually, there's a long list, but that's a whole other show.
Don't ask my wife.
So I mean, I think that's one of the biggest things that's going to happen is it's going to put some of these things, it's going to put them on the back burner, and they're going to be thought of the same way we think of, I shouldn't say bleeding and leeches, because I mean, the leeches do work for some things. I mean, talking therapy actually does work for a lot of people, but I think when we can actually fix what's wrong with somebody very quickly, and I think that's going to happen in our lifetime. And finally, I'd just like to say that my plan is to live forever, and so far it's working out perfectly. On that note, how about we go around and thank our guests for being on the show. It was a great discussion, and I'm glad we had it. One of the things that I found after doing the show is that sometimes the hardcore science is easier. So really, because it's technical. You don't have to make a decision
of what you've done.
Oops. Absolutely. Yeah, but hey, we're thinking about it. We have to be thinking about it. If you're doing it, you got to be thinking about it. And that voice you heard is Dr. George Farr. He's a vice president of biochemistry and biophysics at Aromics LLC and adjunct professor in Department of Physiology and biophysics at Case Western. Thanks, George, for coming on.
You're welcome. It's always good.
And on the same frame, we have Dr. Marc Griswold, Associate Professor in Department of Radiology at Case Western and University Hospitals. Thank you again for coming on the show.
Honor to be here, Marc. Thanks.
It's a lot of fun to have you guys on. Let's go to our remote cameras. How about Dave Brodbeck? Good job.
Thanks,
thank you, Dave. That's Dave Brodbeck, Associate Professor and Chair, Department of Psychology at Algoma University in Sault Ste. Marie.
Always a pleasure, man.
Thanks for coming up. Also, we have, let's go to the next Dave. How about, Vincent Racaniello. Okay. Professor of Microbiology and host of This Week in Virology. And he's also a professor at Columbia, is Professor of Microbiology at Columbia University in New York City. I encourage everybody to go check out This Week in Virology. If Futures in Biotech ever had a related show, one that, I think has a great crossover, it's This Week in Virology. So I encourage everybody to go check it out.
No doubt about it.
It's really fun, and viruses are awfully scary and awfully powerful. So thank you for coming on.
Viruses are us, Marc. Thank you. It was great. I always like being on, and look forward to having you back on TWIV.
Cool. I mean, we are half virus, aren't we? No, was it 9 or 10% of our genome is dormant virus?
Yep. It is what distinguishes humans from chimps are viruses.
Yep. Wow. Oh my God. Okay. So yeah, we're 0.1% different from chimps anyway, right? Or I guess
somebody did some serious engineering a bunch of years ago, eh?
Yeah.
Let's not get into that one, George.
That'll be for the next show. Someone has, yeah. So lastly, we have Dave Thomas, professor and chair of the Department of Biochemistry at McGill University and the Canada Research Chair in Molecular Genetics. Thanks for coming on, Dave. It was really a pleasure and good,
Thanks,
good
Marc.
to talk to you.
That was a lot of fun. We should do it again. Okay.
We will. We definitely will. Thank you. Thanks, everyone, for coming on. I'd also like to thank Burke McQuinn for handling the audio and video boards today and doing an awesome job of switching. We've had 6 people on the show, and this is the largest we've ever had, and he did a great job. I'd also like to thank our new producer at This Week in Tech, Eric Lanigan. He's taken over our initiative over in Petaluma, California. So thank you, Eric. I'd also like to thank the team that make this possible. Leo Laporte, Dane Golden, Lisa Kensel, Tony Wang, Frédéric Louis, Mike Taylor, John Salanina, and Ken Shepardson in Petaluma, and the rest of the team. Thank you for helping get Futures in Biotech on the air. Lastly, I'd like to thank Phil Pelletier and Will Hall for the opening and closing themes. For Futures in Biotech, I'm Marc Pelletier.